Good morning, guys. Hope you guys are doing good and staying safe. Today, I'm here to present my seminar on bioaerosols and the sampling techniques. The contents are general introduction, bioaerosol classification, assessment of air quantity using the principles of sedimentation, impaction, impingement, sorption, and filtration. Principles of bioaerosol collections, and the last one will be the biological analysis. First, we'll study about the basic definitions. What are aerosols? A suspension of a solid or liquid particles in a gas that includes both the particles and suspending gases. Example here. Um, what are bioaerosols? And aerosols of a biological origin, the particles of the biological origin suspended in the air. In outdoor air, 30% of the all particles are larger than 0.2 mu appear to be biological origin. Particulate matter, the generic term for the broad class of physical and biological diverse substances that exist as a discrete particles in liquid droplets or solid forms in the air. Next, we're studying about the biological classification. Depending upon their biological characteristics, bioaerosols can be classified into following types: viruses, parasites, and in the living organisms, the bacteria and the fungi, parts of products of organism that will be the fungal spores, pollens, endotoxins, allergens. Based on the particle size and natural background concentration of aerosols, the types of bioaerosols, the virus, the size will be 0.02 to 0.3 mu per meter. The concentration will be nothing. And in the bacteria, the size will be 0.3 to 10 mu per meter. The concentration will be 0.5 to 1000 meter cube. And the fungal spores, the size will be 0.5 to 30 mu per meter. The concentration will be 0 to 1000 meter cube, and the pollen, the size will be 10 to 100 mu per meter, and the concentration will be 0 to 1000 meter cube. And this we're studying about the viruses. Viruses are intracellular parasites that can produce only inside a cell. The naked viruses ranges from 0.02 to 0.3 mu per meter. Most of the airborne viruses or the part of a droplet nuclei are attached to the another particles with a wide range of sizes. Airborne viruses are transmitted by the direct contact. Aerosolization of the virus may occur by coughing, sneezing, or talking under the favorable ambient conditions. Example in temperature or humidity, etc. Most of the viruses can be served for weeks or months. Next, we'll studying about the bacteria. The bacteria are single-celled organisms with the size from 0.3 to 10 mu per meter. Several factors about the bacteria includes bacteria is generally spherical or rod-shaped. Bacteria often occur as a clusters or chains under the ambient condition. Bacteria colonizes the water or soil and releases as a aerosol when the water or solid is distributed. In indoor environment, bacteria can survive and colonizes the most environment such as ventilation system and become the aerosolized by the air current or vibrations. In addition, bacteria can be classified into two groups based upon their ability to retain the crystal violet dye. Gram-positive bacteria that can retain the dye. They have made up of cell walls that can consist mainly of peptidoglycan, which can cause the many health issues. And gram-negative bacteria that cannot retain the dye. They have cell wall that contain an outer membrane composed of lipopolysaccharides lipoproteins, and another complex micromolecules that can cause health effects. Specifically, endotoxin in outer membrane of gram-positive in gram-negative bacteria are chemically stable and heat resistant. Endotoxins are released when the bacteria cells are lysed and well maintained and their biological activity even after bacteria cells are no longer visible. Example, E. coli and Salmonella. And the fungi. Fungi is a unique group of organisms occur as a single-celled organism. Example, yeast has a multicellular branching structures. More than 70,000 have been identified. Most common fungi dispose by the releasing spores into the air. Fungal spores often occur as individual parasites. The pollen. Pollen grains are produced by the plants to transmit genetic material or near the spherical particle that can range from 10 to 100 μm, which between 25 to 50 μm. The Anaphyllus wind pollutants. The plant produces the ambient bioaerosol. They causes the allergic decreases the airways, hay fever. Pollen forms a variety of common examples, sunflowers, morning glory, and etc. And the next one will be the aerosol factors influencing airborne infections. Particle size that will be lesser than 5 droplet nuclei from coughing and the sneezing. Disposing site depending upon particle size, chemical composition of bioaerosol particles, and the relative humidity. Desiccation, loss of moisture, and the temperature, generally greater inactivation at higher temperature, sunlight, UV inactivation of microbes, factors influencing the hair movements, that is winds, currents, economical hair handlers, seasonal factors, hair current precipitations, and the pollen sources, etc. Hair pollutants, chemically inviting hairborne microbes, open hair factor, enhancing their ability to cause infection in a host. And the aerosols and the respiratory deposition. Aerosols that will be lesser than 5 microns in diameter are removed in the upper respiratory tract, especially the nose. The particles are brought to the pharynx by their activity of the upper respiratory epithelial mucosa. Deposition efficiency in the lower respiratory tract is 50% for the particle 1 to 10 micron diameter, and they can also deposited in the lower respiratory tract, especially the particle will be greater than 0.5 microns, and the particle deposited in the lower respiratory tract can be phagocytized by the respiratory microphages. The next will be the bioaerosol sampling. Many devices have designed for the collection of bioaerosol. Choosing an appropriate sampling devices is based on many factors such as availability, cost, volume of hair to be sampled, mobility, sampling efficiency, and the environmental conditions under which sampling can be conducted. The another factor must be taken into the account, especially when the sampling efficiency of devices. And the factor is related to the maintained by microbial viability during and after sampling. In this session, several types of commonly used sampling methods are sedimentation, impingement, impaction, suction, and filtration. First, we'll study the basic method. We usually know about this method because we did it in first sem. That will be settle plate technique, the hair sampling method for microbial testing that we already done in our labs. The number of methods can be used for the test here for the modes and another microbial contamination. One of the oldest testing here for the microbial contamination is settle plate method. Though the method is semi-quantitative, it can be still considered as a useful method in industries such as food, paramedical, cosmetics, and the method is used to assess likely the number of microorganisms depositing on the product or the surface in the given time. This method involves opening and exposing the petri plates containing agar medium with the suitable growth of microorganism of interest. If one of interest is testing for the mold, the agar plates containing malt extract agar supplemented with some antibiotic to express the bacterial growth could be used. The agar plates are left at the top of the table at the selected area for half an hour to 4 hours. This allows the more spores and the fragment can settle on the agar media by gravity. And this will be the results of the settle plate technique because we already did it, we know about it. And the number of microorganisms deposited on the agar surface of the plate over the period of exposure is determined by the incubation of agar plates at 25 degrees Celsius for 5 to 7 days and counting colonies that can be developed. The result can be expressed as the number of colony forming units per unit time. The counted colonies can be further characterized by genera or the species. The higher number of the higher number of CFUs and or presence of potential of pathogenic such as the Aspergillus species. Advantages. This method is extremely useful for assessing air contamination by microorganisms. It is very easy to conduct and the cost efficient, effective. The advantages. Only the visible organisms that can be detected by this method, hence it may give the false impression that hair is clean if the most of the hairborne microorganisms are dead or fall negative may obtain from the building with a very restricted mold growth and very still hair is undistributed room. Species of poorly culturable modes. Modes can be consist of the species with the poor airborne dissemination. Example: Cladosporium, unpainted cold hair vents, and the Fusarium and many other wet spored fungi. Assessment of air quantity using the principles of sedimentation is the simplest device used for sampling particles larger than 10 micrometer. The collector consisting of the plastic jar is about 20 to 35 centimeter height and 10 to 15 centimeter diameter at the base, which the slight inward wrapping of the walls from top to bottom, and the sample is deposited over the period of 1 month and the material is dried and the wetted. As usually only water insoluble dust fall reported in mg per centimeter cube. The advantages: easy to use, easily sterilized, highly biological sampling efficiency in the comparison with the another sampling devices. Any type of liquid can be used. Liquid impingement media subsamples can be used for testing a variety of microorganisms by standard culturing and the molecular method. The disadvantages: hair cannot be filtered. At the lower temperature, bioaerosols can be embedded because the liquid matrix is solidified. Flow rate of air is equals to 12.5 liter per minute. The media is equal to 0.85% of NaCl and 1% of peptone are media for interest. Next one is impaction. Impaction was developed by the Ertile A. Anderson in 1958. The Anderson six-stage impaction sampler provides the accurate particle size discrimination. It operates at input air flow rate of 28 to 28.3 liter per minute. The general operating principle that air is sucked through the sampling and the port and sticks on the agar plate, and and the larger particles are collected on the first layer. Each successive stage collects the smaller and smaller particles by increasing the flow rate. The shape of the Anderson sampler does not confirm the size of the human respiratory tract, but the particle size distributions can be directly related to the particle size. Initially, Petri plates have the larger pore size so that the large bioaerosols can be trapped. Separation is based on the size of the bioaerosol. Media is of our interest. Advantages. Anderson 6-day sampler is the particle size determination can be obtained, reduces the potential and prevents the sample loss, allows the direct quantitative analysis, enumeration of the fungal spores with a low culturability, time improvement collection method. And the disadvantages is expensive. Biological sampling efficiency is somewhat lower because the method of collection, which is impacting on the agar surface and the microorganism that can be grow directly on the agar surface. Impingement. Impingement is a trapping of the airborne particles in a liquid matrix. The glass impinger operated by drawing the hair through the inlet that is similar to the shape of human nostril passage. The hair is transmitted through the liquid medium where then the air particles became associated and subsequently trapped. Sorry. The impingers as usually separate at the flow rate of 12.5 liter per minute at the height of 0.5 meter with the average breathing height of humans. The impingement trends to very sufficient for the particle in the range of 0.8 to 15 μm. The as usual volume collection of media is 20 ml and the typical sampling duration is approximately 20 minutes. The another feature of the impingement is the process is the liquid and the suspended microorganisms can be concentrated or diluted depending upon the requirement of analysis. Advantages: easy to use, portable, easily sterilized, and high biological sampling efficiency in the comparison with many other sampling devices. Any type of a liquid matrix can be used. Liquid impingement media subsamples can be used for testing a variety of microorganisms by standard cultural and the molecular methods. Disadvantages: hair cannot be filtered. At the lower temperature, bioaerosols can be embedded as because the liquid matrix is solidified. Flow rate of air is equals to 12.5 liter per minute. The media is equals to 0.85 meter NaCl and 1% of the peptone are media of our interest. The centrifugation. Centrifugal samplers uses the circular flow pattern to increase the gravitational pull within the sampling devices in order to deposit particle. The denser bioaerosol deposit at the bottom, and the first row. These samples are able to sample a wide range of air volumes that will be 1 to 400 liter per minute depending upon the size. Polyvinyl chloride is a filter on the top and the air inflow is long time lighter bioaerosols also settles at the bottom. Advantages: relatively inexpensive, easily sterilized, and the portable. Disadvantages: it lacks the highly biological sampling efficiency and the particle sizing capacity. Analysis is performed by the sampler with the elute amount, collection of the elute, and the subsequent assay by the standard methodologies. Suction. Vacuum sampler compresses a pump which is used to draw a air through the nozzle held close to the surface. The suction action generates the combination of lift forces and drop forces, and the particles are removed when through the combination together with any mechanical mechanical forces resulting a large particle set in the motion by the heat stream exceeds the addition forces between the deposited particles and the surface. The air intake is formed by the region between the nozzle and the surface. However, in the most vacuum samplers, the nozzle is close to the surface that the physical of the surface by the nozzle may occur at this provides the additional mechanism by which the surface contamination is dislodged. Advantages: the suction sampling devices have been widely used for the number of inward surfaces sampling applications due to their many advantages, includes the ability to sample the large areas and the portable for constructing the suction sampler by the modifying the existing devices and the suction sampling methods of thermal exposure assessment has been found in unsatisfactory related to the another sampling method such as wiping, and the disadvantages. Intra comparison on the studies is difficult at the laboratory. Laboratory studies are based on the variety of surface contamination methods, and the field studies involves the wide range of surface types and the particle loadings levels. Less particle in the case of suction sampling. The next and the last one will be the filtration and the deposition. Filtration and the deposition method are both widely used for the microbial sampling because of the cost and the portability reasons. Filter sampling requires a vacuum sources that involves the passage of air through the filter where the particles are trapped. Membrane filters can be variable sizes that can tend to restrict the flow rate. After collection, the filter is washed to remove the organisms before the analysis. Filtration sampling for microorganism is not highly recommended because it has a very low all over the sampling efficiency. It is not portable. However, in many cases, low cost makes it is attractive method. Here the fibers are polyvinyl chloride used for the filter, and the deposition sampling is for the easiest and most effective method for sampling. Deposition sampling can be accomplished mainly by the opening on agar plate and exposing it to the wind, which results in the direct impacting gravity settling and another deposition forces. And the problems with this method sampling are low all over sampling efficiency because it relies on natural deposition, no defined sampling rate or particle sizes, and the uh, in- and the intrastatic difficulty in testing for the multiple microorganism with a varied growth condition. Analysis of microorganism collected by deposition sampling is similar to impacting sample analysis. Advantages: filtration and deposition methods are both widely used for microbial sampling because the cost and the portability reason. One case where the filtration is routinely used in the sampling for airborne eh, lipopolysaccharides and the superior collection in uniformity, low sensitivity to the environment condition, sample suitable for the variable and non-variable variable analysis. At disadvantages, the deposition sampling can be mainly by opening by an agar plate and exposing it on the results in the direct compaction, gravity settling, and another disposal forces. The problem with the method of sampling. All low overall sampling efficiency because it relies on natural deposition, no defined sampling rates or particle sizing. The testing and it is used in the mini testing from multiple and it is used in the very less testing difficult, and it is used very difficulty in the testing of multiple microorganisms with a varied mm, growth conditions. The principles of aerosol collection. Inertial impaction of the particle forces the impaction onto the solid or semi-solid impaction forces a cultural medium on the surface that can be examined microscopically. The application of the principle in bioaerosol sampling that include single stage impaction, the surface heat sampler, cascade impactor, two or three impaction stages, slit sampler. The impaction stage consists of two or more slits instead of one or more holes. And the next one will be the centrifugal inertial impaction, the particle separation by centrifugal force in radial geometry. And the liquid impingement, the particles are collected by inertial compaction into a liquid or particle diffusion within the bubbles. The tangential impinger collects the particles by inertial impaction and centrifugation, filtration, impaction, interpretation, diffusion, gravitational settling, etc. The particle physical properties filter force, size, aeroflow, gravitational settling at the least effective methods for biological collection, the particle size, the shape, and the airflow, the deposition of the particles, and the last one will be the electrostatic precipitation, all over some of the physical damages that is caused by impactor and impinger. And the last topic will be the biological analysis. Many different assay methods to detect the quantify the microbes in the collection of bioaerosol samplers are described in the literature. The traditional method for biological analysis that includes microscopic counting and the cultural based analysis. Some of the more advanced methods are currently available include biochemical and molecular biological assay. The first one will be the microscopy. Bioaerosol particles are collected on the glass slide, tap on the appropriate filter, and they can enumerated using the microscopic methods. Large particles, for example pollen grains, are readily enumerated using the light microscope. However, the smaller bioaerosol, for example bacteria cells, they may be masked by another particles, and some smaller bacteria cells may not be visible using the optional microscope. At the certain cases, the collected bioaerosols can be stained to better distinguish them the background material that can be enumerated using the conventional light microscopic methods. Some of the commonly used microscopic techniques that includes the bright field microscopy. The resolution of the field microscopy is zero point 0.2 miometer. That it is time consuming and very expensive. Usually, in which like bacteria or the fungi or the virus will be used mostly under the bright field microscope is Salmonella and Cladosporium. And the next one will be the electron microscope. Uh, Electronically magnified images can be seen in this, and the in we already studied about the electron microscope. We used to see the bacterial structures, the like what and all will be the inside that we can watch, and the like different viruses like morphological studies and all will see under the electron microscope. And this having a very short wavelength, the magnification is up to 22 lakhs x. The example in this electron microscope that we usually watch is hepatitis A virus and the influenza virus. This is viability assay. The bacteria and the fungi agar plates. The non-selective are plate count agar and R2A agar. The selective EC agar, Salmonella sigilla agar, malt extract agar. In the biological analysis, the morphological evaluation and the viability assay methods for the organism in our various media are classical techniques for detection of the bioaerosols in environmental hair samples and the biochemical athletic methods for detecting and identifying microorganisms measure certain microbiological molecules in bioaerosol particles to distinguish the biological and non-biological materials. And biochemical method uses the following high-end techniques or rapid measurement for specific chemical properties of the microorganisms collected. That will be gas and liquid chromatography, mass spectroscopy, fluorescence and luminescence spectroscopy, and the flow cytometer and electrochemical techniques. And the references are here where I referred, and thank you.